Science is a method of discovery. It's given us the tools to shape our civilizations, build the frontiers of tomorrow, and redefine the limits of our imagination. At the heart of these achievements lies the tireless efforts of research scientists whose labor preserves the noble spirit of discovery. This show celebrates scientific research traditions by exploring exciting new innovations in science and technology. As we continue into the 21st century, we will once again look to science to meet the challenges ahead. This is the spirit of research. This is the tradition of discovery. This is the method.
Scientists today. If the universe had a beginning, in the beginning there was only darkness, water. Can you hear me?
E is equal mc squared.
Why are we here?
In which energy is Z equal to mass.
Where did we come from? The human race was improving in knowledge and technology.
We be converted into a very large amount of energy.
Flying is rough. All those long hours waiting at the airport, the stopovers, don't even get me started on the jet lag. But for some birds though, it's just a part of their survival. Some do it for a short distance. Others go far, far longer. The bar-tailed godwits, for example, flies over 11,000 kilometers to their destination, non-stop. I get tired from just flying an airplane for just a few hours. Yes, bird migration remains one of the greatest spectacles in the natural world. But how does it happen? Why does it happen? Researchers are beginning to finally unravel the mysteries of this natural wonder.
From a human perspective, when we think of these tiny birds flying 10 or 15,000 kilometers annually, we imagine that this must be some incredible marathon, something that exhausts them, and they're living on the edge, traveling such amazing distances. But from the bird's point of view, they're such efficient flying machines. Their whole physiology is designed to power this kind of migration. Their wings and bodies are designed for efficient flight, and actually they can travel these distances quite easily.
Bridget Stutchberry is a professor of biology at York University. She specializes in tracking birds during migration.
The main purpose of migration is so that animals can better survive by switching environments. So if you think of Canada and these small little insectivorous birds that I study, the little songbirds, they can't stay here in the wintertime because there's no food. And so they have to leave. A bigger question is, why did they come here in the first place? And it turns out that Canada and the northern US is a perfect place to breed. Days are long, there's tons of food. It's really easy to raise a family. It's just that you can't stay here all winter.
This lifestyle is not without its challenges. Studies have shown that one of the most dangerous times for birds is during migration.
One of the reasons we're so interested in tracking the migration routes of these birds is to discover the key stopover sites and habitats that they need to complete their migration.
Most birds don't fly nonstop. They have to rest and refuel.
With all the changes that have happened to their landscape with agriculture, cities, etc., it's very likely that we're inadvertently causing mortality through wrecking their stopover habitat and stopover sites. But we've never been able to map out where these birds stop and what regions are most important for them to stop and refuel.
One of the biggest challenges is that most migratory birds are under 20 grams. And you can't use global positioning satellites to track them because the devices simply aren't small enough.
There are several techniques to get around this limitation. Ryan Norris is a leading researcher in the study of bird migration.
One of the things we commonly use are chemical markers to track where individuals have been the previous season.
These chemical markers can be found in metabolically inert tissue such as feathers.
So the way it works is that if we have an idea of where a feather is grown, we can sample that feather the following season and say, this individual likely grew this feather in this location the previous season.
The food and water that birds consume contains isotopic signatures that are unique to their region. Scientists can determine where the birds have been by analyzing the isotopes found in the feathers.
The isotope technique is non-invasive. You're taking a small feather sample from a bird, so it doesn't really affect their flight patterns or their foraging ability.
Chemical markers can't give you pinpoint accuracy. They can only narrow down the bird's location to an area the size of Florida.
Another external device we can use on small birds are geolocators.
Geolocators are light sensors, and they gather data on light levels, every 10 minutes, 24/7. So you might wonder, well, why are we measuring light levels if we want to know the bird's position? From those light levels, we can determine the time of sunrise and sunset. And on any given place on the planet, if you know the date and the time of sunrise and sunset, then you can figure out where exactly your bird was, within about 100 kilometers.
Bridget and her team were the first to use geolocators to track small songbirds.
We've been putting geolocators on 2 different species of songbirds, the purple martin, which is a type of swallow, and the wood thrush, which is a forest bird. To get the geolocators on the birds, we first have to catch them with a nest trap or a mist net. These geolocators, they fit on the bird's back, and you just slide it up one leg and around the other leg, just like a kid would do putting on their school backpack. After we've put it on, the feathers, the back feathers, will cover up the actual geolocator itself, and all that shows is the tiny little light stalk.
Unlike GPS devices, the data from geolocators needs to be manually downloaded.
Getting the geolocators back, it uses the same sort of methods as putting them on in the first place. The key is that you have to be able to predict where your birds are going to come back to. So with purple martins and wood thrushes, they tend to come back to the same breeding territory year after year. So you can go back and catch them. And then once they're in your hand, you can take the little backpack off and get your geolocator back.
These trackers were originally designed by the British Antarctic Survey to track larger species of birds.
Over time, other researchers wanted to use the same technology for, let's say, geese or ducks or hawks, so they made them smaller and smaller and smaller. So this weighs only 0.6 grams, and it is the world's smallest geolocator at this point.
Considering the size of the songbirds, does the added weight of the geolocator affect their ability to migrate?
We can't find any differences at all in terms of birds that wore the geolocator versus ones that did not, in any measures of health or performance or survival or anything. But as far as we can tell, it's kind of like us wearing a ring or a watch or glasses. They don't even know it's on their body, I don't think. So when we see purple martins flying 400 or 500 kilometers per day with the backpack on, we wonder, well, if it's slowing them down, then how fast would they be going otherwise?
Geolocators and chemical markers are effective tools that help researchers track small migratory songbirds. But why is it important to study this behavior in the first place?
Well, one of the main reasons to track migratory songbirds, aside from their amazing migration behavior in marathons, is that unfortunately so many of these species are in steep decline.
Migratory birds are huge players in ecosystem functioning and ecosystem services for Canada and the US. They keep insect populations down, they disperse pollen, and they're food for a wide range of other types of animals.
There's a real urgency for trying to understand not just how they migrate, but what places they need to stop in order to refuel and have a successful migration. Those are the habitats that need conserving. So now with these geolocators, we can actually map out the migration routes of different populations and different species and piece together what are those critical bottlenecks that we need to protect to make sure this migration system stays intact.
The longest migration is 71,000 kilometers, 2 times farther than what was previously thought to be the longest migration. That's like running the Boston Marathon over 1,600 times. And it's done by this little guy, the Arctic Tern, using a zigzagging path to go from Greenland to Antarctica each year. What's more incredible, the bird only weighs about 4 ounces.
When scientists study the effects of a drug on patients, they'll likely have 2 groups of test subjects. They'll give one group the actual medication, and the other, nothing more than a sugar pill, a placebo. Placebos are one type of scientific control, and they're very important to the research process. Scientists understand that people's perceptions can deceive them, and controls are a way of curbing that. After all, we've all heard the expression, your mind is playing tricks on you. And anyone who's seen a good optical illusion can attest to that. It also makes science a more reliable method of uncovering truths. It's science's way of confirming the absence of evidence is not evidence of absence. And what we're seeing is evidence of what is actually there. Our five senses: smell, sight, hearing, touch, taste, are our brain's way of interacting with the world. But sometimes, those senses betray us. Don't believe me? Okay, watch and listen carefully. Ba, 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 ba. Now keep watching, ba, and listen carefully. Ba, 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 ba. Did you hear something different? Ba. Because I said the same thing both times. Ba. Watch and listen again. Ba, 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 ba. This is the McGurk Effect. Ba. It's a very vivid example of how your brain can deceive. Ba. Your brain is hardwired for memory to connect the particular movement of my mouth to that sound. So when you hear me say fa, even though the sound coming out of my mouth is ba, your brain is telling you to hear fa. This means that what you are hearing is actually what you're seeing. Ba, 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 ba.
Unlike most optical illusions, the McGurk Effect illusion does not break down even after you understand how it works. However, studies show that the listener's culture and native language can affect how intensively the McGurk Effect gets received. Dutch, Spanish, German, and Italian-speaking listeners seem to experience the strongest McGurk Effect, while Chinese and Japanese-speaking listeners are the least susceptible to the illusion.
What do you think of when you hear nanotechnology? You have heard it, right? Ah, nanotechnology, the newest science buzzword. The world is nano this and nano that, but what does it mean? Nano refers to the nanometer. It's a unit of measure for really small things. How small are we talking? Well, I'm about 2 meters tall. My fingernail is about 1 centimeter long. Now let's go smaller. The freckle on my face is 1 millimeter, while the diameter of a hair on my chin is 100 micrometers. A red blood cell measures about 5 micrometers. And a DNA strand is just 2 nanometers. Now we're in the world of nanometer. Nanotechnology is simply the manufacturing of materials and devices at this scale. Now, it may seem small, but doing this has big advantages. Many science disciplines have already found the benefit in going nano, including the medical field. Cancer is a devastating disease. Research and development for cancer treatment and detection is one of the most pressing medical frontiers of the last several decades. Cancer becomes very difficult to remove when it spreads throughout the body, a process called metastases, and it is often treated with chemotherapy.
Chemotherapeutic drugs are so potent that often you don't die of the cancer itself, but you'll die of the side effects of it.
Molly Shoykat is a professor of biomedical engineering at the University of Toronto. She specializes in developing targeted drug delivery systems to treat cancer and other diseases.
The advantage with chemotherapeutic drugs is that they're highly toxic. The limitation is that they kill both healthy cells and cancerous cells.
That's not desirable. That's the reason why people get sick when they undergo chemotherapy. Nanotechnology can allow us to deliver anti-cancer agents in brand new ways.
Karine Ho is a researcher working with Dr. Shoykat. Their work involves using nanotechnology to improve targeted drug delivery systems to treat cancer.
Because those cancer cells are growing and dividing very quickly, in order to grow to a larger size and be a really well-established tumor, they need to recruit their own blood supply.
The accelerated growth decreases the quality of the blood vessel structure. Unlike normal healthy organs, the tumor's blood vessels are full of small holes.
Because of these gaps, nanoparticles, although they're small, they're still large enough that they don't typically go through normal blood vessels, but they will go through cancer blood vessels.
This phenomenon is known as the enhanced permeability and retention effect. By loading chemotherapeutic drugs into nanoparticles, scientists can target tumor cells with greater accuracy.
So a drug molecule is very, very small compared to a nanoparticle. By taking these very small drug molecules and repackaging them into nanoparticles, that size difference blocks them from being able to enter normal tissues as easily as they would normally.
This is called passive targeting. Like a sieve, nanoparticles accumulate in greater numbers near tumor regions because of their size.
Active targeting involves taking a nanoparticle and putting bioactive molecules on the surface that are able to recognize and bind to cancer cells.
The nanoparticles that Molly and her team developed are attached with an antibody. These antibodies target and bind to antigens that get overexpressed on some types of cancer cells.
Because we're interested in breast cancer, we've been working with an antibody called Herceptin, which is used clinically. And we've been taking Herceptin and chemically conjugating it to our nanoparticles. And inside our nanoparticles, we have the chemotherapeutic.
About 25% of breast cancers overexpress an antigen on its surface. These antigens are actively targeted by the Herceptin attached to the nanoparticles.
So Herceptin itself has a therapeutic benefit. So what we're trying to do is take advantage of not only the therapeutic benefit of Herceptin, but also its ability to target specific cancer cells that overexpress those receptors.
One of the other important aspects of active targeting is that once a nanoparticle is bound to the surface of a cell, typically what will happen is that that cell will then take that particle and internalize it, bring it into the core of the cell. The reason why this is important is that most drugs, most anti-cancer drugs, in order to function, need to be inside the cell. And so active targeting, while adding another layer of selectivity, also brings the drug to where it needs to be in order to be effective. And so the overall goal of using nanotechnology to deliver chemotherapeutics is to get another layer of selectivity so that more of the drug will go to the tumor instead of going elsewhere in the body after injection.
Nanoparticles may also benefit other areas of cancer treatment besides drug delivery.
So there's a whole new area called theranostics. And what theranostics is it takes advantage of therapeutics and diagnostics.
Nanoparticles let the idea of theranostics really come to life, particularly if we remember that we can have a vehicle that can deliver a drug and almost the exact same vehicle deliver an imaging agent.
David Jaffray is a renowned medical physicist. His work focuses on developing and improving imaging systems for diagnostics.
There are many different ways that imaging is used, and anything we can do in the context of imaging to be more sensitive, so to detect things that are small earlier, or to be more specific. In other words, I don't just see something, I see something and I know what it is. Anything we can do to increase what we call the sensitivity or the specificity of the imaging test is very valuable. Nanoparticles have a promise of increasing the sensitivity. For example, if we could have them target a small cluster of cells and have that cluster of cells show up brightly on an imaging test where those cluster of cells are cancerous cells, that would clearly be useful.
Increasing the sensitivity can make cancer cells easier to detect. It may also allow doctors to determine the most effective way to treat the cancer.
The treatments for cancer right now work fairly well, but they're not perfect. They don't work for everybody. And new tools are allowing us to identify that tumors for individual patients are slightly different. But we're also learning that even the tumor's ability to take up a drug, for example, varies from patient to patient. So now we're starting to learn that it's a possibility for us to actually measure for each individual patient how well the drug is being accumulated. And if we know that, we could know that some patients would do well with one drug or not, and we can then optimize or choose the best therapy for that individual patient.
I don't know that we can achieve all of this with polymeric nanoparticles or nanotechnology, but that's certainly the goal, and that's what we're working towards, is trying to do a better job of killing just the cancer cells.
Though the technology is not yet available, scientists speculate that one day nanorobotics will eventually be used to repair cells and even treat diseases. These won't be the robots you're probably thinking of. They would be synthetic biochemicals with specialized functions in the body, kind of like white blood cells, bacteria, or viruses.
Science continues to be integral to our modern civilization because we recognize it as an engine to our economic stability, our inherent need to explore, and our quality of life. Research in the field or in the laboratories is a tool scientists use to unravel the nature of our world. Sometimes human applications of a particular research are not always clear or immediate, but nevertheless crucial to the overall scientific endeavor. Science is not an end, it's a continuum. It's the best method we have devised as a species to innovate and create in this world. Thanks for joining us. See you next week on The Method.
Scientists today. If the universe had a beginning, in the beginning there was only darkness, water. Can you hear me?
E is equal mc squared.
Why are we here?
In which energy is first equal to mass.
Where did we come from? The human race was improving in knowledge and technology.
Maybe converted into a very large amount